Hello and welcome everyone to this podcast for epigenetics. In this podcast we're going to focus on histone modifying enzymes. In the last set of podcasts we talked about the different ways that a histone could be modified. To remind you, we talked about histone acetylation, histone methylation, histone phosphorylation, histone ubiquitination, histone summation, histone ADP ribosylation. All of these modifications covalently added a specific molecule to the histones. Adding a new covalent bond requires a great deal of energy. So what we want to do today is talk about these enzymes that do this act. And we're going to focus primarily on histone acetylation, histone methylation, and histone phosphorylation. I'm going to say just a little bit about histone ubiquitination and the enzymes involved, but not much. And I'm not going to talk about summation or ADP ribosylation as far as the enzymes that are involved. So let's get started here with histone acetylation. So histone acetylation. Histone acetylation is one of the most positive marks on histones, particularly H3, that suggests active transcription. So if we have our DNA here with our histones attached, like so, with their internal tails, if the gene underneath these histones, underneath these nucleosomes, is going to be actively transcribed, we know that at H3 tail, it's going to be acetylated. And probably not just at one site along that H3 N-terminal tail, but it's going to be heavily acetylated. However, that acetyl group here wasn't always there. So let's remove it right now. And for now, I'm going to focus here on H3K9. If you remember from previous discussions, this is the one that competes with methylation and can be a key trigger, an on-off switch, determining whether or not that gene will be transcribed. The enzyme that is involved here in adding that acetyl group is called histone acetyltransferase. We call it HAT, H-A-T for short. And it will come along here and add this acetyl group to it. So how does it add this acetyl group? Well, let's go ahead and draw kind of this generic looking enzyme. And you'll notice I drew two active sites, or rather two binding sites on this enzyme. The first one here is the active site that will bind H3K9. It'll bind this lysine. Now remember, we want to add an acetyl group to this lysine. What better way to do it than to have an acetyl group sitting in this other active site? So what sits here is a molecule called acetyl-CoA. This is a common strategy, having the molecule that you want to add to the substrate sitting right here in an alternative binding site, so that it's in close proximity with this lysine here that it needs to be added to. Now remember what it's doing here. Well, it's doing a few things after it adds this acetyl group. For one thing, lysine has a positive charge. By adding this acetyl group here, you neutralize this positive charge. It masks it. This will likely loosen its association with the DNA, but it also prevents these N-terminal tails of neighboring histones from interacting with each other. Now, so as we're talking about these histone acetyltransferases, I want to mention that there's not just one acetyltransferase. There's not one histone acetyltransferase. There are several within a genome, and they have specific targets. They don't all bind to H3K9. They may bind to this one, but they may bind to another target. Some may have multiple targets. In fact, most do. There are also some acetyltransferases in the cell that don't associate with the histones. Those would just be acetyltransferases. But this is a very common enzyme activity within the cell. But specifically with HATs, those acetyltransferases that acetylate lysines on the histones, there are three major families, and I want to mention them just real briefly here. The first is MYST. The next one is GCN related N-acetyltransferase, often just called GNAT. NAT. And finally, this last one, P300. And within each of these families, there are many members. They all have slightly different specificities, as I said before, and different targets expressed at different times, different kinds of affinities. Now, once we've added acetyl groups to each of these, and gene transcription is now cruising along, there has to be a mechanism set in place to be able to turn these genes off. And one of the mechanisms involved is removing the acetyl groups here. Now, as you're learning throughout the semester, it's not just the act of adding one acetyl group that turns this gene on. It's not going to be the act of removing the acetyl group that turns it off immediately. It's a whole cascade of events, but we have to start somewhere. And so we're just talking about this one event. And by removing this acetyl group, that does help bring about other changes that will eventually shut down expression here. So the enzyme that removes this acetyl group is called histone deacetylase. We often refer to these as HDACs or HDACs. And what they will do is they will have this enzyme bind to this acetylated lysine here and then break that bond, removing this acetyl group from here, making this now just H3K9. So I'm going to erase this, and this acetyl group, this HDAC here is now off of this area. And if you remember, just as a bit of a review, this H3K9 with the acetyl group removed will now be likely to be methylated. And having that methyl group there will now begin to shut down this whole process. There are 4 classes of HDACs, and the names aren't incredibly clever, but I'll go ahead and list them. It's 1, 2, 3. I hope you're sitting down because the last one is surprisingly 4. Each one of these classes, just like the different families of the hats, the different classes of the HDACs include many different members with different kinds of roles and specificities. Okay, that's all I'm going to talk about right now regarding histone acetylation. I want to now move to histone methyltransferases. Alright, so histone methyltransferases. And at the end of this topic, I'm going to talk about histone demethylases, which remove methyls. But let's just stick with the addition of the methyl groups for now. And we might as well draw our familiar DNA here with our histones attached. I should say our histone octamer attached. It's not just one histone. Remember, it's the complex of 8 histones that form the nucleosome. And we can draw our histone tail here, like so. And we might as well stick with one of our favorites here, maybe 2 of our favorites here, K9 and K4. We've talked quite a bit about these 2 over the past few days. They play a pretty important role in gene regulation and epigenetics. Something we don't mention enough, and we probably should mention it more, is that when things are acetylated or deacetylated or methylated, these aren't typically transient events. They can remain methylated or acetylated for many cell divisions, thereby remembering, so to speak, to keep these genes on or off. So what methyltransferases do is they will come along here to one of these lysines and they'll add a methyl group. Now I say lysines because that is one of the major methyltransferases. So let me list just real quick the 2 kinds of methyltransferases. The first are the histone lysine methyltransferases. And all this time that we've been talking about methylating these lysines in one way or another, we've been talking about this class of enzymes, the histone lysine methyltransferases. There is another class that we'll spend a little bit of time talking about, but not a ton, but we do need to give it some more credit, are the histone arginyl methyltransferases. But I want to stick right now with the histone lysine methyltransferases. And to give myself a little bit more room, I'm going to rewrite that up here. I want to write it up here so we don't forget that that's what we're talking about. The histone lysine methyltransferases. Let's begin talking about these histone lysine methyltransferases. And one thing that we need to mention is that all of these histone lysine methyltransferases have a acet domain. The set domain is the catalytic part that transfers the methyl group. So I'm going to start building this complex over here, and this is going to be set. These set domains are found in all eukaryotes and in some bacteria. In bacteria, the set domains aren't going to be found in histone lysine methyltransferases because there are no histones, but they are associated with enzymes that are involved in transferring methyl groups. In eukaryotes, these usually have a specificity to H3 and H4 histones. Now over here in our enzyme here, I'm going to make some changes. So remember, this is our set, like so. And I'm going to make 2 binding sites here. This binding site will be for the lysine amino acid that's going to become methylated. This binding site binds a molecule called ADO-MET, ADO-MET. Just like we saw with the acetyl-CoA being close to the lysine in the histone acetyltransferases, in these set domains found in these lysine methyltransferases, in these set domains we have the molecule that's going to be added to the lysine right next door to it. This increases the possibility of being able to transfer this MET to this lysine. It lowers that activation of energy. Now I want to talk about some set containing proteins. That a protein complex that contains this set domain. Your book mentions several of them, but I want you to just know a few of them. The first one is called set 7/9. It monomethylates H3K4. So this set 7/9 can only take in one methyl group. This particular enzyme has the capacity to open up and allow a second or third methyl group to come in there, but in doing so it distorts the active site in such a way that it can't add that second or third methyl group to the lysine. Right, the second set containing protein is EZH2. And this one di and tri-methylates H3K27. So we haven't talked about lysine 27 yet, but this is just another methyl group that can be methylated to affect transcription. A third one here is SUV39H1. It also di and tri-methylates, but its target is H3K9. Okay, number 4 here is GLP. GLP mono or di methylates H3K9. So you can see how possibly GLP must act first in order to bring in the second or the third methyl group to lysine 9. Okay, before we move on, I realized I didn't mention something about set 7 and 9. Remember, I said it adds a mono methyl group to H3K4, and I said it couldn't add a second or a third. So there must be another enzyme that can add a second or third methyl group to H3K4. And that's the one I want to talk about next. And I'm going to have to erase this so I have the room. But I wanted you to recognize that MLL1 could possibly work in concert with set 7/9. So this is our 5th example, MLL1, and it can tri-methylate H3K4. MLL1 got its name because of its link to myeloid and lymphoid leukemia. When this receives a mutation that translocates this gene in front of another promoter, it can become constitutively active and it can lead to these leukemias. It has some other roles too, and it turns out to be a very important gene. In fact, it's one of the very early genes that's necessary for embryonic development because it's a master regulator of Hox genes. If you haven't read about Hox genes, you should. These are a really fascinating set of genes that are important in setting up the body pattern of various organisms. But MLL1 is the one gene that helps regulate these sets of transcription factors, these Hox genes. To kind of talk about how it does this, we need to talk about some of the other proteins that bind to this set domain of the MLL1 complex. So I'm going to draw it over here. So we have another protein that binds to this complex around here called WDR5. WDR5 acts to mediate the association between H3K4 and the methyltransferase complex. And when I say methyltransferase complex, I'm talking about the set domain here. So WDR5 is able to identify these locations. It brings in the set domain here to carry out this catalytic event of adding a methyl group to that lysine. There are two other proteins involved here. The first is a protein complex called RBBP5. RBBP5. This stands for retinoblastoma binding protein. Retinoblastoma is a tumor suppressor gene that this protein associates with in other contexts. Within this complex here, I'm not aware that it actually binds to retinoblastoma. It might, but I don't think it does. Sometimes proteins get named something because of the way it was first identified. But then we discover that a protein may have more than one function or may associate with other complexes, such as in this case, it associates with this set complex. And we believe it has a role in the structure here, holding the structure together, so a structural role, and helps to activate set. Alright, there's one more protein I want to mention, and that is ASH2. Now ASH2 has been a long known regulator of Hox genes. Perhaps ASH2 in this complex helps MLL1 find these Hox genes. Now we know some things about this, about the roles here, that these three elements when attached to set are important. Because we know that if WDR5 is mutated, this results in no methylation. And we know if RBP5 or ASH2 are mutated, RBBP5 is mutated, that we get reduced methylation. And same thing with ASH2. It results in a reduced methylation. I'm running out of room here, but I did want to say one thing about ASH2 here. And that is, it's got a link to a human disease. And this human disease is called Kabuki syndrome. It's a very rare disease. It's something like 1 in 32,000 live births have this syndrome. So it's not very common, but it leads to various abnormalities found in children, including mental disabilities. Okay, that's all I want to say about these set domains. I now want to spend a little bit of time talking about some methyltransferases that don't have a set domain. So these are going to be called non-set dependent methyltransferases. So as the name implies, when you look at the structures of these, they don't have one of those set domains in it. And I'm going to talk about just one of these non-set dependent methyltransferases, and its name is DOT1L. It has a unique role in that it doesn't methylate something in that N-terminal tail, but rather it methylates histone 3 lysine at position 79. And this is part of the core. It's on the outer edge of that core, but it's still in the core, so it's not on that tail. It was first discovered in yeast, and one of the few things that yeast epigenetically regulates is the telomere. And so usually the telomere is silenced, but in mutations of .1L, the telomere is not silenced when .1 is mutated. Now, like the other enzymes that we've talked about, it also has these two binding sites. One of them that takes in adelmet to provide that necessary to transfer. And over here would be that H3K79 binding site, with that same idea of keeping that methyl group close to the substrate where it's going to attach to. That's all I want to say about this nonset dependent methyltransferases. Alright, now I want to spend just a little time talking about protein arginine methyltransferases. So up to this point, we've been talking about protein lysine methyltransferases. We know a lot more about the lysine methyltransferases, but we also know that the arginine methyltransferases are important. So it's equally important that we talk about them for at least a little bit. We don't have time to spend a great deal of time on them, but I want to introduce you to them. The one I want to talk about is this one called PRMT1 for protein arginine methyltransferase. Now, I want you to know this particular enzyme, and these are the few things I want you to know about it. First, you should know that, as the name implies, that it is an arginine methyltransferase. Now, there are lots of these methyltransferases in the cell, and we know most about this one because it's a very predominant one. About 85% of all arginine methylations are caused by PRMT1. Doesn't mean it's the most important, it just means it does a lot. And lastly, I want you to know that PRMT1 knockouts, so when we remove the gene from the genome, these are embryonic lethal after implantation. So shortly after it implants, the embryo dies. Next, let's talk about lysine-specific demethylases. So let's remember that adding a methyl group to the lysines and the arginines, but we're just talking about the lysines now, that this reaction is reversible. So this set domains are important in taking this lysine residue and adding a methyl group to it. And then methylases can reverse that reaction, removing the methyl group and restoring the methionine and restoring the lysine with its positive charge. The one methylase that I want you to know is LSD1, which stands for lysine-specific demethylase. So if we were to draw our DNA here with our histones on it, like so, with our N-terminal tails and our H3K4s, and remember, when these have methyl groups on them, that is a mark to increase transcription. So if we wanted to turn off gene transcription, one way that we could begin to set that stage is that this LSD1 enzyme can remove these methyl groups. Now, interestingly, it can only remove mono or dimethylations. It can't remove a trimethylation. Another enzyme would have to be involved in removing the trimethylation. Now, interestingly, this reaction that removes these methyl groups here is a rather toxic reaction. Going from lysine with methyl on it to just a lysine generates hydrogen peroxide and formaldehyde. Hydrogen peroxide is a reactive oxygen species, or can be. As a reactive oxygen species, it can damage DNA, proteins, membranes. It's a very nasty thing to have in the cell. The cell can accommodate that. It can remediate it, but it's still not something you want to have a lot of in the cell. Formaldehyde is a known mutagen. Formaldehyde is known to cause mutations. And the tricky part about all this is this reaction is occurring where? It's occurring in the nucleus, where there's a lot of DNA. And we're generating ROS and this mutagen that could easily mutate our DNA. This strategy to turn off a gene must be worth it. There must be a way in the nucleus where these byproducts, these toxic byproducts, are remediated and gotten rid of. But it is an expensive task just to shut down a gene. Not only is there energy involved in removing the methyl group, but there is energy involved in repairing any damage that might be caused by this, and energy involved in removing these toxins. Okay, so next I want to talk about an arginine specific demethylase. And I'm not going to ask you to know too much about this one. I would just want you to know its name, which is peptidyl arginine deaminase 4. Or we'll make life easier and we'll call it PAD4. So its job in a cell is to remove methyl groups from arginines. More specifically, it removes it from histone 3 R8, R is for arginine, histone 3 R17, histone 3 R26, and lastly histone 4 this time, not 3, histone 4 R3. That's all I want you to know about this particular enzyme. Okay, the next enzymes I want you to know about are those that are involved in phosphorylation of histones. And to remind you, there are two types of enzymes involved in phosphorylating and dephosphorylating. They're kinases and phosphatases. Kinases add phosphate groups. Phosphatases removes phosphate groups. Now, if you remember in our discussions, we spent our time with phosphorylation of the histones talking about H3S10. So we had our DNA over here and our histone proteins here with our end tails like so. H3S10 would be here. So the kinases that bind to this H3S10 site and add a phosphate group to this serine fall into one of three types. So there are three types of H3S10 kinases. The first is one we call ribosomal S6 kinase, or RSK. The second one is MSK1 and MSK2. And the last one are these Aurora kinases. And I want to spend a little bit of time talking about the ribosomal S6 kinases and the Aurora kinases. So these ribosomal S6 kinases. These kinases have been shown to phosphorylate H3S10 in response to cell division, survival, cell differentiation, and a fun one here, migration behavior. Ultimately, what they respond to, and I should say initially what they respond to, are usually some kind of growth factor that interacts with a cell and then sends a signal to various downstream pathways that will ultimately activate the RSK. And then it will have an effect on these four and other activities. Mutations in RSKs can lead to this disease that's called Coffin-Lowry syndrome. Okay, now I want to talk a little bit about that third class, those Aurora kinases. Aurora kinases have been studied for a very long time, and they've long been known to be involved in cancers. And we know that these aurora kinases will phosphorylate their targets during specific times in the cell cycle. So to remind you, G1, S, G2, and then our M phase. And we know that aurora kinases phosphorylate S10 beginning at late G2, so in this area right here, and then through metaphase found during mitosis. It will also phosphorylate H3S26. Now I want to mention one more kinase, and this is a kinase that is not linked to H3S10, but it is linked to something I mentioned during our last couple podcasts, so I want to come back to it. And this is called MST1, and it's involved in phosphorylating H2B-S14. And when H2B-S14 is phosphorylated, it leads to apoptosis. If you remember, when we were talking about the effects of phosphorylation, I did mention that there was some evidence that phosphorylation was linked to phosphorylation events on the histones that led to apoptosis. So I wanted to make sure that we mentioned the enzyme where that link exists. Alright, we're almost done. There's just two more enzymes I want to mention, and I'm not going to say a whole lot about them. The first is the opposite of phosphorylation. It's the removal of the phosphate groups. And that's going to be the role, as I mentioned before, of the phosphatases. So these will remove phosphate groups. And there are two that we could talk about, and we may come back to later, but for right now I just want you to know these two classes. The first is cysteine dependent phosphatases, and these are often just called CDP. And the second class are these metallophosphatases. So all I want you to know about the phosphatases as far as their role in removing phosphate groups from histones is that they do have a role in removing phosphate groups, and that there are these two classes. That's all I want you to know about phosphatases for right now. Alright, the last enzyme I want us to think about are the ones involved in ubiquitination of histones. And all I want you to know for this, at least for now, is that the enzymes that add ubiquitin to the histones are called E3 monoubiquitin ligases. I want you to know two examples of these monoubiquitin ligases. The first is ring 1 and BMI 1. These work together to make sure that ubiquitin is added, one ubiquitin molecule is added to these histones at the appropriate sites. Okay, that ends this podcast. If you have any questions for me at all, please make sure you stop by. If not, I'll see you in class.